Have been looking at certain aspects of the nervous system. We've been talking about sort of the microscopic level. We've talked about neurons, we've talked about the channels that are on these cells. And so what we're going to do here is sort of zoom out and look at the big picture in terms of the layout and the anatomy of the nervous system. So we understand neurons, right, so far to this point, how they are the main communicating cells that transmit certain signals that are responsible for a whole host of functions, like breathing, like the beating of the heart, like the movement of skeletal muscle and smooth muscle and cardiac muscle. But what we're going to speak about now are some of the other structures that are in the nervous system. What are the supporting cells? And we'll talk about how their functions help to support that communication on neurons. All right, so our first objective is to describe the different types of neurons and supporting cells and then identify their function. So the central nervous system is made of the brain and spinal cord. Everything else outside, so all of the peripheral ganglia, all of the peripheral nerves are going to be considered peripheral nervous system, and we'll talk about those in the upcoming unit. So what we'll focus on is the brain and its spinal cord. Function is maintaining homeostasis, right? So as we've said before, it's going to help to detect stimuli. It's going to send those changes up to the main processing center, right? The medulla oblongata mainly. And then it's going to send signals for certain corrective changes. And so that is sort of that loop that is important for homeostasis. So as such, the brain and spinal cord play an important role in terms of maintaining homeostasis. There are tons of neurons and even more so synapses, right? So we talked about the fact that you can have convergence and divergence. So one neuron can receive many different axon terminals synapsing onto it. Likewise, one axon terminal can synapse onto several different spots on a single neuron. So there are lots of neurons, about 100 billion, and even more synapses as they connect with each other. The brain is responsible for everything that we perceive, everything that we do, everything that we feel and think. And so it is understood to be a large part of our physical anatomy, right? And our functions, but also a large part of our personality, right? So our behavioral patterns, our thoughts, our memories, our feelings, all of that can be perceived and understood via the nervous system in the brain. All right. Having said that, however, the brain is still one of the least understood organs in the entire human body. It's still a lot of unknown regions or unknown functions that are associated with certain regions. And so it's still an area that's ripe for research and better understanding how various parts of the brain really relate to their functions in bringing about our personality, etc. Let's talk about some of the players, some of the structures that are involved. We've got neurons, which are the main signaling cells. Okay, we talked about the structure of a neuron. It's got a soma, an axon, and then the axon terminal. We've also got some supporting cells called glial cells, and we'll talk about different types of those. So there are 5 different types of glial cells, and we'll define them. The brain is also going to be a physical support for the central nervous system. Okay, so it's going to be kind of the house where all of that processing takes place and then signals can be sent and received. The blood supply to the central nervous system we'll also talk about, and then the blood-brain barrier, as well as the difference between gray and white matter. So these are some of the features of the central nervous system, and we will get into each of these. Okay, let's start out by looking at neurons. So they are the structural and functional units of the nervous system. They're going to respond to chemical and physical stimuli, right? Whether that's touching something or perceiving a certain chemical smell or the sensation of food in the gastrointestinal tract, we can detect those biomolecules. So neurons can perceive various types of stimuli and then conduct these electrical impulses. That is how we send and receive those signals. There's also going to be a release of chemical regulators, right? The neurotransmitters that are part of that signal transduction or signal transmission. And so it enables us to perceive sensory stimuli. It enables us to learn things, to to have memory, right, to remember certain concepts. It's really interesting. It's always funny when we think about using the brain to understand the brain. So it helps us do a lot of our daily functioning, as well as some of the more physiological functions as well, like gastrointestinal secretions, like salivary gland secretions, like our reproductive and endocrine hormones. All of that is controlled through the neurons and the nervous system. Now, neurons cannot divide, but they can repair themselves. So this is why when there's nervous injury or spinal cord injury or even injury to the brain, it's really hard to get that part of the brain, specifically the neurons there, to regenerate, but they do have the potential to repair themselves. So if there is any function that comes back, it's usually going to take a much longer time as those neurons slowly repair instead of just dividing and creating a brand new cell. That's not possible. Okay. In the frame here on the right, we can see what a neuron looks like, right? We can see lots of different synapses happening onto the soma here. All right, so here's our soma in blue. Here's our axon, the length of our axon. Here are several different axon terminals synapsing onto the soma itself, right? We've also got things like myelin. So in the last unit, we looked at saltatory conduction and the way that myelin was interspersed along the axon. And so that myelin, which is essentially fat, just helps to insulate the neuron and help to speed up that electrochemical transmission. Now in the periphery, we can see a few other types of cells, and these are the cells that we're going to describe later on, but notice they're closely related to the neurons, and as we'll see, they also help to support the neurons. They're going to do things like make the myelin, or they're going to also help to clean up the area, right? Clean up any debris that's around the neurons. So those can be considered the glial cells or the supporting cells. Okay, continue on with neurons. We can classify neurons based upon their structure and function. So on that, we can think about the number of processes that extend from the cell body. And so on that basis, we can think about unipolar versus pseudopolar or multipolar neurons. Or we can classify neurons based upon their function. So what is their job or their role in terms of perception? So if they have more of a sensory perceptive role, they would be sensory neurons. Or if they're responsible for transmitting motor instruction, right? So going to your skeletal muscle, they would be considered motor neurons. And then we also have a subclass called interneurons, which can help to bridge that gap. So in between a sensory neuron and a motor neuron, we have an interneuron that can help to transmit that signal. So in other words, there are several different ways to classify neurons depending on exactly the focus or the aim. So here are some examples. We're going to focus mainly on the classification from a functional perspective. So we've got sensory neurons that conduct impulses from the sensory receptors up to the central nervous system. So this is an example of a sensory neuron. It's going to pick up stimuli via receptors and then transmit those chemical signals to the brain and spinal cord. So this will help us detect things like sight, right? The retina can be considered a sensory neuron. The olfactory or the neurons responsible for smell and olfaction, those can be considered sensory neurons. The vestibulocochlear nerve can perceive hearing via the vibrations in the ear, and then those can also be considered sensory neurons as well. So they're going to bring in information from the world around us. Motor neurons are going to conduct impulses from the central nervous system in the opposite direction, so out towards the target. And this can be muscle, which is the main case, but it can also be glandular tissue as well. So it can send signals to skeletal muscle, right, giving instructions to move. It can send signals to smooth muscle. Think about blood vessels, think about your gastrointestinal tract, or it can send signals to cardiac muscle, which is the heart. And then we've also got glands that can receive that signal as well. So motor neurons are then going to be divided into things like somatic neurons. Somatic neurons go only to skeletal muscle, or autonomic neurons go to the other types of muscle, smooth muscle, cardiac muscle, and then glands. So we can further divide motor neurons depending on exactly what type of muscle they go to, or if they go to glands. And then the association or interneurons are sort of in between, right? So here, there's no example shown here, but in between a sensory and a motor neuron, right? So for example, here, we would have a smaller neuron that would be considered an interneuron. So if I drew in a little neuron connecting this neuron, right, to this neuron here, this would be considered an interneuron, bridging that gap between motor and sensory. And this is sometimes involved in things like reflexes. So reflexes will perceive, like if you touch something hot, without really going to your brain cognitively, you'll move your hand right away, right? That's an example of an interneuron function. It's going to go from motor or from sensory right to motor without really involving the brain. Okay, let's talk about the supporting cells now. So the supporting cells are actually the majority of the cells that are in the nervous system. So it's a smaller portion of neurons themselves, and then a larger portion of cells that help to support and clean up the area in the brain and help to support those neurons. So glial cells are going to be non-excitable, so they're not involved in action potentials. They're not involved in necessarily transmitting signals. They are not synapsing the way that neurons are. They are more playing a supportive role as opposed to actual signal transduction. So in this image, we can see lots of the different types of neurons. We can also see the division between the central nervous system and the peripheral nervous system. All right, so here's the division that shows us that the brain and spinal cord, which are on this side of the image, really are where the somas or the cell bodies of the neurons reside. And then notice how the axons project all the way out to become the nerves, which are out in the peripheral nervous system. Okay. Also, we can differentiate the neurons themselves, which are here in yellow, right, just extending across, from all of the various types of supporting or glial cells. So we have ependymal cells here. These are the cells that are going to make cerebrospinal fluid. We've got things like astrocytes. Astrocytes are shown in green. These are going to help to extend and bridge the communication between the neuron and the blood vessels. They help to form the blood-brain barrier. We'll talk about what that is. We've also got things like oligodendrocytes, which help to make the myelin in the central nervous system. And then alternatively, we have Schwann cells, which help to make the myelin in the peripheral nervous system. Okay, and then the fifth type is going to be the microglial cells. So let's go through each of those cells and talk about their function a little bit more in detail. So astrocytes have a lot of different functions. As I mentioned, they can bridge from the brain or the neuron, excuse me, to the blood vessels and help to protect what gets to escape the blood and enter the nervous tissue. So the astrocytes help to really protect, they sort of guard the nervous tissue and make sure that there aren't any pathogens, any viruses, bacteria that are escaping the blood vessels and moving out into the tissues. Ependymal cells, on the other hand, line the cavities in the brain that are filled with cerebrospinal fluid. Microglia are going to be phagocytic cells, and then we talked about oligodendrocytes and Schwann cells. Okay, so kind of going over the same concepts here. Astrocytes are star-shaped cells. So that's going to be one easy way to pick them out. They're going to bridge, as I mentioned earlier, this connection. So here's our blood vessel. Here is our neuron, and then here's this astrocyte sort of acting like a gatekeeper from, preventing certain compounds or chemicals or pathogens from escaping and entering the nervous tissue. They also help to modulate synaptic activity. So we talked about this in terms of things like presynaptic facilitation or inhibition. So astrocytes can also encourage the neuron to release either more or less neurotransmitter. So we talked about that in the previous topic, that presynaptic facilitation was encouraging the axon terminal to release or dump more neurotransmitter, whereas inhibition was preventing it from releasing or dumping neurotransmitter. So in that way, astrocytes can encourage or can have an impact on the synapse, but remember, they are not synaptic cells. They are not excitable cells. They don't have an action potential. They can just help to encourage, by way of calcium release, how much neurotransmitter is released at the junction. They also help to communicate through chemical messengers as well. So they can send certain messengers like glutamate that will encourage the terminal to do different things. Other things that astrocytes do: remove neurotransmitter from the synaptic cleft, so help to eliminate or clear some of those neurotransmitters from the junction. They help to maintain a steady electrolyte composition in the brain interstitial fluid, so the fluid that's floating around the central nervous system. They help to regulate the amount of electrolytes in that fluid, make sure it's at a healthy balance, especially as it pertains to potassium levels. The astrocytes also protect neurons. So they protect neurons, as I mentioned earlier, really by forming that, that bridge, or that bridging that gap in between what gets to leave the capillaries and escape into the nervous tissue. And so by way of doing that, they can eliminate toxic substances, oxidative stress, and protect the brain and neurons in that way. Astrocytes can also secrete glycogen, which is one of the main sources of fuel for the brain. So the brain is very unique in that it can only use glucose. So it's very glucose dependent. And so having an ability to make glycogen, which can then be converted to glucose for energy, is one of the ways that astrocytes help to support neurons. Microglia. So microglia are also helping to protect from toxic substances. They're going to play a more active role in that protection by actually engulfing bacteria. This is called phagocytosis, which is where the cells can essentially eat other components in the tissues or in the cell. So they can engulf bacteria, they can engulf old or dead or dying or injured cells, and they can protect the brain from oxidative stress or toxic exposure as well. Clinical correlations here to make as far as glial cells are concerned. We've got things like multiple sclerosis. Multiple sclerosis is a loss of myelin. So the two cells that are responsible for making myelin are the oligodendrocytes in the central nervous system and then the Schwann cells in the peripheral nervous system. So in any of those instances, if there's a loss of the amount of myelin that is on the axon, that can lead to multiple sclerosis, which is where there's a loss of brain functioning, usually starting with vision and muscle weakness, and then progressing to things like respiratory failure as you lose the ability to send nervous impulses to the lungs and to the heart. So multiple sclerosis is one example of how we can have a clinical syndrome because of the loss of myelin. Alzheimer's and Parkinson's are two other clinical diseases that we see that are regarding glial cells. Alzheimer's is specifically a loss of cholinergic neurons, so neurons that release acetylcholine, and that is typically replaced by scar tissue. So you'll typically see the brain shrink in size as we lose a lot of that that nervous tissue, and that is because we simply lose neurons. We lose the amount of neurons. In these patients, we can see things like cognitive decline, where they have an inability to understand or perceive their surroundings. They can forget who they are. They can forget their family members. So that can be quite impactful on their quality of life, as you would imagine. And it's all coming from the loss of the neurons that are responsible for sending the signals that help us live a normal life. Parkinson's is similar to Alzheimer's, but here we have a loss of specifically dopaminergic neurons, as well as some of the glial cells that produce inflammatory agents. So here for Parkinson's, the main difference is we have now a movement disorder. Dopamine is one of the neurons or neurotransmitters that helps us to have a smooth pursuit, right? So when I reach out my hand to grab something, that happens in a very coordinated and smooth way. If we lose dopaminergic neurons, there's going to be a very a shaky maneuvering or a tremoring or a trembling whenever we try to move or do things. And so there's not that smooth pursuit with actions. And so that is how Parkinson's will typically differ from Alzheimer's, but they both will have the cognitive decline, the loss of memory, etc.